everyone, my name is Maddie, and I'm a wildlife ecology apprentice at the Wilds. Today I'll be talking about my research into burying beetles. Burying beetles, genus Nicrophorus, are a family of insects who use small vertebrate carrion as food for their larvae. Nicrophorus exist over a wide range, including the majority of North America. Some burying beetle species that have been recorded in North America include an orbicollis, N. pustulatus, N. tomentosus, and N. americanus. N. americanus, also known as the American burying beetle, has been on the endangered species list up until very recently, October 15th of this past year to be exact. The decline of the American burying beetle, which is quite a mouthful and will be addressed as ABBs from now on, is believed to be correlated with the extinction of the passenger pigeon. There are current wild populations of ABBs in Oklahoma, Rhode Island, Nebraska, and in Ohio. The Wilds is a 10,000-acre safari preserve in southeastern Ohio. The Wilds actively participates in various wildlife conservation efforts, one of which is an ABB breeding program. ABB breeding has taken place over the past 4 years at the Wilds, with breeding stock growing each year. Currently, the Wilds has bred and released 3 generations worth of burying beetles. To monitor the success of this breeding and release program, 23 pitfall insect traps are positioned around the Wilds property. These traps have been used not only to monitor the ABB population upon release, but also to monitor other burying beetle populations that live on site. And every year since the beginning of the breeding program, more burying beetles of varying species have been captured on Wilds property than the previous year. In order to more accurately assess the success of this breeding program, this experiment was designed. In this experiment, several abiotic and biotic factors at each trap site were compared to see if one or more significant factors influencing how many beetles were collected could be isolated. Because the ABB population is small, the other burying beetle populations who are on site, who fill similar niches as ABBs, were used to assess how ABBs may be acting in the same environment. Over the current 4-year breeding and release program, data was recorded of each burying beetle captured. The individual was identified by species, and the site where they had been captured was noted. If the beetle was an ABB that had been captured previously or released, its recapture was noted. Analysis of each capture site was broken into 2 scales, patch and landscape. In the patch analysis, elevation, wind exposure, and vegetation percent cover at 1 and 3 meters above the trap were recorded. Wind exposure was measured using a percent difference using 2 Kestrel 3000 handheld devices over 1 minute. One device was held inside the trap area while the other was held outside. Vegetation percent cover was estimated over a 1-meter radius circle, with the center being the trap. The landscape analysis consisted of land cover percent estimates within a 1-kilometer radius around each site, and distance from the site to the nearest freshwater source. ArcGIS Earth and the National Land Cover Dataset were used to estimate the land cover percentages, while ArcGIS Earth was used to measure distance from the site to the nearest observable freshwater source. In the middle of the poster, you can see a map of the Wilds with the trap locations labeled. The red points are the sites where recaptures occurred, the white points are the 2 release sites, and the blue points are all of the other sites. Next to it are the factor measurements for each site, just for your reference. Analysis of this data was done using Python regressions to create best-fit linear models. Python regressions are general linear models that are commonly used when working with count data as the dependent variable. The value returned by a Python regression in R is called the AIC, or Akaike Information Criterion. This value indicates the strength of the model, but only in comparison to the other models using the same regression and data. The lower the AIC is, the better the model fits the data. The analysis was broken into 2 parts again, total beetle capture counts and just the ABB recaptures. Remember, the goal of this experiment was to use the larger populations of burying beetles as possible indicators of how ABB will act. However, we do have a small sample of American burying beetles who were released by the Wilds and then recaptured. This sample size is too small to reveal any significant findings, but a separate analysis was run just for general consideration. To begin, a model was created for every combination of factor measurements at the sites run, with the total beetles captured at these sites over 4 years. Running singular factors through the regression returned percent grassland cover and percent forest cover as the most significant indicators of beetle captures, with percent grassland being a direct relationship, while percent forest was indirect. The combination of factors returning the lowest AIC value was grassland, percent cover of vegetation at 3 meters, wind exposure, and percent cover of freshwater. The chosen model was only the best fit based on the other models run, so to find out how well it truly fit the data, a normal QQ plot of the residuals was created. This first QQ plot can be seen above the wilds map. The closer the QQ plot points are to the normal QQ line, the better the model fits the data. Looking at our QQ plot, there is a noticeable deviation from the normal line in the residuals points, specifically one positive standard deviation away from the mean. The same methodology was used for the recapture analysis with a few adjustments. 2 more factors were added as independent variables, distance from release site and distance from Zion release, and only the site counts where recaptures occurred were used. Because all recaptured beetles came from one of the possible 2 release sites, distance from these sites were considered as a possible indicator for recapture. Once again, all possible models were created and compared to find the model with the lowest AIC value. The best fit model had freshwater proximity, vegetation percent cover at 3 meters, distance from release site, and freshwater percent cover as significant factors indicating beetle capture success. Another QQ plot was created from this model's residuals, and it can be seen below the map. And as you can see, this QQ plot had even greater variance than the first. The residuals of the 4-year count data QQ plot, the one above the map, vary greatly from the normality line, especially after moving 1 positive standard deviation away from the mean. This indicates that even though the model was the best fit out of the models we created, it's not a strong indicator overall of beetle capture numbers. And the same can be said for the second QQ plot with recaptures. The variance away from the line of normality is greater than the first QQ plot. This could be due to a much smaller sample size of only 63 beetles, or it could just be a weaker model overall. Considering this, we can assume none of the measured factors are strong indicators of beetle capture success. Applying this study to the wild's breeding and reintroduction program, there's no need to move or relocate any of the current traps solely based on the environments around them. None of the measured factors cause significant increase or decrease in the number of beetles caught, meaning that the wilds, and possibly other breeding and reintroduction programs, don't need to worry about things such as elevation or wind exposure when choosing locations for their burying beetle traps. Areas of further study at optimizing trap locations could look at beetle movement across a landscape from a single release point. This could possibly find areas of avoidance or areas of preference that beetles use. Well, thank you for your time and listening to me talk. I'm not sure how the format is going to work, but if you have any questions, please let me know.